with,
Yes, kindly go ahead, sir.
yeah, okay. So it can be thought of as a central hub that unites several disciplines and methodologies. And we can conveniently divide this hub as having a support side and a scientific side. While on the support side of this hub, we have all this information technology, how you manage information, how different software applications, various databases, and all the computational resources, whether it's a hardware or software, that provides the infrastructure for the bioinformatics. While on the scientific side of hub, we have various methods which are extensively used in molecular biology, biotechnology, pharmaceutical technology, and various other medical fields. Example like genome, protein sequencing, structure prediction, molecular modeling, drug design and development, which is the main reason of why we are discussing this topic here in today's presentation. Now, one of the important aspects of this is how to use bioinformatics in drug discovery. Well, is it the slide is moving?
No, sir, like it is still on the first slide.
Okay.
Yeah, now it has moved, sir.
Good. So computational drug discovery is not a new thing. It is already a well-established scientific discipline in medicinal chemistry research, which is critical in the development of various new treatments. And also it is now an indispensable tool in long process of drug discovery and development. It provides us the options for understanding chemical analysis systems in different ways, and also yields information that is not easy to obtain in laboratory analysis with considerably less cost and effort than in experiments. It has created lots of opportunities to speed up and rationalize the multidisciplinary drug discovery process. Okay, let me put it in this way, and provide novel approaches to the design and development of drugs. Well, the main objective of computational drug discovery is the development. At the same time, how you apply these developed methods in integrative bioinformatics and cheminformatics methods that can be translated to support modern drug discovery by designing novel drugs and to improve human health. So earlier, we used to do molecular modeling using plastic models, not so distant past, at least when we were studying biophysics or doing undergraduate studies with Professor T. P. Singh at AIMS almost 25 years back. We used to study big models while understanding molecules. We used to have these plastic models. I'm sure many of you have also studied build models using these plastic models, where you have different color coded spherical balls representing different atoms, and these are joined by different cylindrical rods, which gives you the various bonds. And this is how you build various models and study the properties of the molecules. These were essentially physical models. They were based on the molecular connectivity and about the stereochemistry that was associated with these molecules. They gave some impression about the molecular dimensions and also about the shape of the molecules that you were modeling. However, they were not so accurate, and also they were not they are difficult to manipulate. Suppose you have a set of these resources and you want to build molecules around this. You make a sum. You made a sum wrong. You put some wrong atoms or a molecule in place. Then everything is the whole model goes wrong, and you cannot keep on making these models and storing. So you have a limited resources. You have to rebuild models by the same set of the resource that you had in it. Often fragile instantly, very easily they can fall on the ground and break. You have very little information about the energy and their properties. So in a way, the chemical representation was more or less very limited in this, in these type of physical models that we use to model. And these were good for building models for small molecules. If you want to build a model of a large models, these were not very handy and had a limitation. Now, thanks to all this advancement in the computers, both in hardware and software, today we rely mostly on computers for the molecular modeling exercise. Molecular modeling, this allows access to molecular information, which can be used for predicting properties and how these can be used for various applications in drug design, drug discovery, and various other applications. Whether you want to see the properties of a small molecule, whether it is a chair or board conformation, whether how a molecule can be built together to have a various properties, or see how a molecule, large molecules are assembled together to form a assembly, how a protein or DNA interacts, and so on. So there are lots of applications which you can easily study using these computer-aided approaches instead of having been used by the plastic models. Now, not long back in 2013, if you remember, the Nobel Prize for Chemistry was awarded to these three gentlemen named by Michael Levitt, Martin Karplus, and Ariel Warshel. If you remember, they say that the chemical experiments ideally takes place in labs, but the ability to carry them out in a real time through computers has won these three gentlemen this Nobel Prize for taking chemistry to the cyberspace. So, what does that mean? This highlights the importance of how important the computers are there in current era for studying various properties of molecules. Their research in 70s help us to understand how exhaust fumes are purified or how photosynthesis takes place in green leaves. And this kind of knowledge makes it possible to find the best design for things like new drugs, solar cells, or catalytic converters, or for cars. So there are lots of applications. Today, the computer is just as an important tool for chemists as a test tube. So the idea of showing this paper cuttings of in the year 2013 is to highlight that the computers have now grown in stature and have played a very crucial role, and this is a very important, and this can't be ignored in the current area of modern drug discovery. So why do we build model in first place? Well, this is true for any field where the three-dimensional shapes of molecules and their properties are important can usually benefit from modeling. This is true for the aircraft builders, whether it's a Boeing or Airbus or Bombardier or any aircraft manufacturer. They simply cannot go right in the workshop and start building their aircraft. So they have to first do a lot of modeling. They have to study each and every aspect, how the aerodynamics is going to work out, how is going to be the wingspan, how his tail is going to be placed, and all these things have to be, they do study all these aspects before actually a single bolt is being fitted in a unit. And this is true for the manufacturers of car or designers of the cars. Also, they do all these things. The design, the model, and before it goes on the floor for being manufactured. Or for that matter, for building a house, you need a model to see how the various apartments are put relative to each other in a township. What is the size of a room, a study room, or a dining room? So all of these things are being modeled first before actually these are being implemented in actual real sense. So scientists use models to see how things work before they actually make the final product. And this is in a sense is a safer and a less expensive. The chances of failure of the project is more or less is reduced. So this is how the model are needed to be studied before actually, and this is now true for the drug designing also, as we progress further during my presentation. Also, if you recall 1996 Nobel Prize for Chemistry was awarded to Professor Harold Kroto and Professor Karl and Smalley for their discovery in 1985 of a new allotrope of carbon in which the atoms are arranged in closed shell, which they call as fullerenes. Now here you can see Professor Kroto with a bunch of fullerene models on his desk. This shows the geometry of fullerenes, and every point of these models represents the location of carbon atoms, and the lines represent the bonds holding two atoms together. So these models have been explaining the various properties of the fullerene. This is again an example of how the model help in understanding the properties and the various structural aspects of any given molecule. Now, generally, modeling usually involves two important aspects. One is computation, which does all the computing for you, and the graphics, which allow you to visualize what are the computation actually did for you. Typically, you have modeling exercise can have prediction and visualization of shape and properties. That means you can predict the shapes and properties of various macromolecules as well as the small molecules. Macromolecules gives you information about the targets for being developing small molecules, the drugs or inhibitors or any, for that matter, the protein ligand interaction studies. You can compare the shape and the properties. Now, suppose you have two molecules, molecules A and B. They have a similar shape, but they probably have a difference in certain functional groups. Now, these functional groups gives a different properties to different molecules, and these properties can be very well studied using the molecular modeling approach. You can examine, predict molecular interactions, how a molecule interacts with a small molecule interacts with a macromolecule, whether it is a DNA or a protein or RNA, how it reacts, what are the interactions, and so on. Investigate stable, unstable, or excited molecules, modeling of dynamic systems, vibration, diffusion, conformational changes, reactions. So you don't have to worry about all these things. Basically, you have to remember that the modeling aids in understanding and prediction of molecular phenomena underlying any given process. So these the computation models, the calculations are done by various taking in consideration of various parameters, and the computer graphics allow you to visualize these models. So once you have a model in place, you can really alter the display. You can change atoms, bonds, color properties of the molecules that you have made. So we see that the spatial representation has definitely improved as compared to the models that used to work with earlier using a physical models or plastic models. They are highly interactive in a sense that you sit in front of your molecule and say that I want to change you one of the residues, say by phenylalanine to tyrosine or tryptophan, or I want to see the effect of how this change makes to the overall binding or of the structural component composition of the. So this is, in a way, it's highly interactive. Molecular models use chemical properties represented mathematically by evaluating equations in computer programs. This all has led to advancement in understanding the chemical property representation by these computational means. Now, so this modeling then has find interaction applications at the interface of whether you are studying structural biology or a biochemistry or medicinal chemistry, toxicology, pharmacology, biophysical chemistry, information technology. So I believe that many of our faculty colleagues who are attending, they are from various backgrounds and belonging to some of these disciplines, and they can try to relate how these modeling methods can be useful for them to engage them in their research work, or can be useful to them for working further with their projects. As we proceed further with the presentation. Quickly, all of you are very well aware with this, but I just quickly try to refresh this information about the protein. Well, because we are going to discuss more of more aspects of the structure of protein in later slides. So I thought maybe I can quickly go through this about proteins. These are actually examples of polymeric macromolecules where the sequence of amino acids give rise to specific structure and function. So this protein folds in 3 dimensions, and structure is organized hierarchically from what we call so-called as a primary structure, which is a sequence of chain of amino acid, and then you have a secondary protein structure. This occurs when the sequence of amino acids are linked by hydrogen bonds, and so you have a beta pleated sheets, alpha helix, coil structures, coils, and so on. And then you have a tertiary protein structure. This occurs when certain attractions are present between alpha helices and beta sheet. And so this gives a sort of order to the protein and the fold. And this is what we are going to further focus on as we proceed further. And finally, you have a quaternary protein structure in a protein consisting of more than one amino acid chain. Also, you have an assembly or a complex of quaternary protein structure. And the structure of the protein has to be studied to understand the function of this, because the structure of a protein is directly related to the protein's function, probably even determining it. And so the reason for the research of 3D structures are the foremost. The reason for our discussion today is in medicine, understanding how molecules function and interact, and this all underpins efforts to develop new drugs in medical treatments and understand the treat and treat diseases. This provides us the targets for finding novel drugs that can block these the functional properties of a protein. Of course, there are other applications in agriculture, while you do protein engineering for better nature crops, or industry where you synthesize enzymes, so on. But we are not at this moment worried about this. So we'll focus on the first aspect, how we can exploit 3D structure of protein in medicine. Now, the details of the protein structures are generally can be revealed through 3 of the biophysical methods. I'm sure many of you must be aware of this. First of them is the X-ray crystallography. This provides the 3-dimensional structure of the target macromolecule, the ligand binding site, how the ligand bind, what is the orientation of the ligand in complex with the protein. So here you use a crystal structure, and X-ray is refracted. You get a diffraction pattern, and electron density map is generated, which is further translated into an atomic model for the studied molecule. On the other hand, you have a nuclear magnetic resonance spectroscopy, another biophysical method. This is also equally important for determining the structure and the functional groups of the ligand, which are likely to be important for binding to the protein. And the third is a cryo-electron microscopy, where you don't use the crystal for studying the structural properties of molecules. And these are useful for solving structures of transmembrane proteins or bigger molecular assemblies, and so on. Based on these biophysical methods, there is another method which we called, and which is also a reliable methods for studying structure, is what we call as a theoretical prediction of 3-dimensional structure of a protein, or we simply call it as a molecular homology modeling based approach. So suppose if you don't have an access to these experimental methods and you have a sequence with you and you want to study the structural properties of these of a given protein. So here these methods can be handy to you. Now suppose you have a sequence protein. If your protein sequence shows a significant homology to another protein of known 3-dimensional structure, then a fairly accurate model of your protein 3-dimensional structure can be obtained by this modeling approach. So what you do here, you use experimentally determined protein structure. You designate as a template to predict the conformation of another protein, which is called as a target, which you call as a target that has a similar amino acid sequence. Now, this is based on a knowledge or of about a template. So you also call as a knowledge-based modeling or a homology modeling. So you start with a protein of unknown structure, you do fold assignment or sequence alignment, and you find if the sequence similarity is very low, maybe you can go for the fold assignment and predict the structure of this. But suppose you find a good homology, a pretty decent identity or similarity between the template and the target, then you can use a structure based template or homology model for building a structure of your unknown protein sequence. The model is built, including side chains, loops. It is further refined and assessed for its quality. Also, the construct models are checked with regard to the conformational aspects and corrected for energy minimized using various force field approaches. So you get a good quality predicted model that can be used, that can be further utilized for various uh, 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 structure-based approaches, as we discussed. Right? So if you look at the relationship between the target and the template sequence identity that you have developed based on the homology modeling and the information content of the resulting homology modeling, then you see as the higher the sequence identity, the more accurate the resulting structure info. So if you don't have any template, so use ab initio methods, that is methods the models are being built without any template, and the quality of these models are generally not good. Uh, a less homology between sequence identity between the target and template, you may use a full prediction methods like threading profile, and the structural content that you get out of these can be good enough for doing some side-directed mutagenesis studies or assignment of protein function and so on. As you go further ahead between the sequence identity and target, between template and target, maybe 30% and more, then you use pairwise sequence alignment or multiple sequence alignment, and you get a better quality of the model that you can derive using these methods. So between these maybe about 50, you if you see the if you superimpose uh, target and the template, you can see a more or less a very good fold prediction, and it overlaps pretty well over this 100%. Between 90 to 100%, see exactly overlapping. That means the model that you have predicted is very, very good, and it completely overlaps on the template. So essentially, the higher the sequence identity, the more accurate the resulting structural information, and then these can be used for various applications in drug discovery, like structure-based drug design, structure-based target assessment, site-directed mutagenesis, assignment of protein functions, and so on. Okay. So one example of how this protein modeling works is one of the respectable protein modeling server that maybe you don't need to have access to very sophisticated program. You simply go to xsysmodel.xpc and you have a protein sequence with you. You can try building your model there. Essentially, it is a set of programs where at each step it does its calculation, like the BLASTP. I'm sure most of you are familiar with this. How do you the way the sequence is analyzed, and similarities between target sequence and the sequence of known structure is calculated, and once the template is identified, simulation is done. The protein modeling generates all the models, and once a model is generated, it is minimized for all the models using various force fields. And at the end, you get a output in the form of various solutions being predicted by the program, which you can use as a model for further studies. Now, once you have a three-dimensional structure, it is useful to look at the protein 3D structures. How do you do it? There are numerous free programs for doing this, including GRASP, developed by Anthony Nichols at Columbia University, or RASMOL, or a Swiss PDVware, or maybe you can go to Google, do the XPECI search, xpeci.ch, and you'll find a whole plethora of programs that can help you in this visualization of various models that can be generated by various other programs. So more spaces with groups studying structural biology have commercial packages like Schrodinger, Sibyl, Discovery Studio, Preset, VLife, which contains more features than the visualization package, which are described in these academic programs like RASP or Swiss PDB or RASMOL and so on. So this is an example of how a RASMOL or a Swiss PDB visualization looks like. It's a representation of a protein DNA complex. So you can see these coiled, the helix and beta sheets, which represents a protein which is bound to a helical DNA. This is the same molecule represented by Swiss PDB viewer, which presents by Swiss Institute of Bioinformatics. And it can provides a very good interface for viewing various models that can be either downloaded from PDB or you can make on your own using a very suitable modeling software. Now, at this point, it's important to mention about one of the very important database, which I'm sure many of you are aware, is what we call as a protein data bank. This is actually one of the best depository of protein three-dimensional structures. More than 180,000 protein structure files have been deposited in this. Files contain text annotations and numerical three-dimensional atomic coordinates. So you can see, so over a period of years, so many structures have been deposited and keep on growing. So currently this stands at around 180,000 total deposits. The structures that has been solved by either X crystallography or NMR or cryo-EM, they actually find their way into this repository. And as a drug designer or a computational chemist or computational modeler, you can download the structure of the protein of your interest from this depository. Simply go to the www.rcsb.org and get your protein from there. You can retrieve these structures either going to the website, as I mentioned in the previous slide, or also you can find various links via the ANTRAs or BLAST links following sequence similarity searches, and so on. Now, also we know that the function of most of the protein is to bind some target molecule or a set of target molecules and perform some action. So one of the basic tenets of pharmaceutical chemistry or medicinal chemistry is that the biological activity of any molecule is dependent on the three-dimensional placement of specific functional groups. And these what we call as a pharmacophore. And understanding this protein ligand interaction mediated by these pharmacophores. And also we know that with only a very few exceptions, the drug mostly interact with macromolecular targets, and these are mostly often with the specific binding sites of membrane bound or nuclear receptors, enzymes, transporters, or ion channel. So this relationship between drug and targets can be explained by what we call as a drug lock and key mechanism. So in this case, the target is actually your protein. The structure of that is being revealed by experimental methods, and the active site is of the target or the lock is being characterized by various computational methods. So you know exactly where is the binding site or the reactive site where you can have a drug or a drug-like molecule that can go and fit in snugly. So most of the drugs snugly fit to their targets, and this is how a drug or a target essentially works. So structures of some locks are available. So PDB is a repository of all these locks. So suppose you want to unlock a particular lock, so you have to first go and find the PDB and see if that lock is deposited there, that particular protein that you are looking for is there. So the structures of some locks are there. These are available from the PDB, but not also. Study of the protein structure provide you the details of the log, and knowing the log structure, you can identify, design the suitable key that can be a good binder to that particular log. This can be achieved by various computer programs. And in general term, we say this is what is the structure-based drug design. So you look at the structure of a protein, or for that matter, a DNA or RNA, and this, the structure from X-ray NMR or modeling, you define the active site, characterize it, and you use different algorithms and software to build a key or identify a key from a bunch of keys which can best fit to the log or the protein structure that you are studying. So the three-dimensional, we have been discussing this structure of proteins. The reason why, because the three-dimensional molecular structure is one of the foundation of structure-based rational drug design. And this offers some of our best hopes for putting an end to many diseases, relies on the, so based on the knowledge of the structure, we can classify them as having a receptor-based approach, where you can use various methods like molecular docking or de novo design or virtual screening to exploit the structure for various approaches. On the other hand, if you have a knowledge about the available potential compounds, you can devise as a ligand-based methods where you can do a pharmacophore modeling or a three-dimensional quantitative structure-activity relationship study to improve the activity of molecules. Or more recently, there has been trends of using the artificial intelligence-based methods, including both the deep learning and machine learning, to improve the activity and efficacy and potency of a set of molecules using these approaches. Now, if you further look at the structure-based drug design, then the idea of structure-based drug design is essentially to identify a better key for a given law. So one of the way to find a better key is what we call as a virtual screening. This is nothing but a computational filtering of a large body of molecules, which is a ligand database. So you have to filter out a set of molecules and identify those binders that have a high probability of activity in a biological system. So you have a ligand database, and you have to find out which of the ligand is going to fit well into the active site of it. So one of the way to graphically demonstrate this by work from my one of my very earlier PhD student, Priyanka. So she developed this model of protein from dihydroorotate dehydrogenase from Plasmodium falciparum, and she used screening. So this is a protein. The active site of this protein is characterized by various important residues, which you can see now. They are popping up here in various colors. And so once the active site is defined, a whole set of molecules are being pumped into this in a sort of computational filtering. So this will rank them on the basis of certain ligand, protein ligand complementary element. In this case, it was the flexes, so it used a certain set of scoring function and then rank them on the basis of how these are fitting well to the protein. So this way, you can prioritize the molecules which have a potential of showing a better activity in a experimental system. So this is how you do a rational way of identifying the molecules from a huge set of library of molecules. Now, you can study how a protein actually interacts in more details by looking at what we call as a molecular docking. This is a very important methods where you explore the interaction that occurs between a ligand. A ligand could be an atom or ion or a molecule or a drug or substrate or inhibitor, which attaches itself to the docking site of another molecule, usually a enzyme or some. Now this interaction between a molecule and a receptor, it is much like a space shuttle goes and docks into the space station. So you can imagine a space shuttle like a Discover or a Columbia or whatever, it goes up into the sky, in the space, And there you already have an International Space Center. So this is, you won't imagine that it will go simply there and get attached itself. There is a specific attaching or docking site designated at the ISS where it goes and finds. So it goes up there, docks at the specific site. Only then there is a specific interaction between sets of astronauts or cosmonauts or whatever. This is in a similar way. You have a protein molecule. It has a designated docking site, and it's only at this docking site that your small molecule or a drug or inhibitor can go and dock, and with the proper affinity it can bind. So by understanding this interplay between ligand and its receptor, the docking site of the receptor, and thus disable the functional properties of the protein and help in designing drugs for disease condition. And ultimately we can use to identify various drugs in most devastating disease. And this, we have seen lots of example currently in COVID-19 intervention, that we have seen lots of molecules have been studied by these methods. Now, this is another example. Not long back, one of the headline that made attention, like crew capsule with dummy abroad docks at the space station. So in this case, the Elon Musk SpaceX, they launched a White Dragon capsule developed by them. It goes up into the space, and then here it was sort of doing autonomously flying. So it does not needed a guiding of a robotics to put it into place. So this is somewhat like a blind docking. It finds its way itself on the docking site and attaches itself without the help of the robotics or any assistance. So as compared to the guided docking, where you define exactly where the molecule has to be placed in a grid, here is a sort of blind docking. You don't define where is your docking site, and you go and find your docking site. And this is how a blind docking programs also work. Now, most of the docking programs basically use the same principles to calculate bound structure. They look for site of interaction between ligand and a target. So Pizarro long back classified them on the basis of the algorithm that has been used, like fast shape matching, mostly used by DOC and UDOC, incremental construction algorithm used by FlexX, Hammerhead, Slide, or Taboo Search, the genetic algorithm, Monte Carlo simulation, distance geometry, some of the programs that is being used. And they use these different algorithms, but basically they use the same principle to calculate the bound structure and looking for site of interaction between the ligand and the target. Now, to give an example of how one of the program, which is also most commonly used by many researchers, because as it comes for academic licenses free of cost and is very useful, is that's what the reason why I have included here. But unfortunately, this animation is not working. This is an example of benzamidine docking to beta trypsin. Anyway, you can go to this site and see the movie there also. So what exactly is this? This is a beta trypsin is a protein, and the benzamidine docking is. So what these guys at the Scripps did, they extracted this molecule from the active site and tried to dock it again with the autodock program. So each frame in this animation would have represented the final confirmation of a separate autodock. It is not running, otherwise you would have seen a flipping of this molecule in the active site of this protein. So each of this run began with the ligand in a completely random translation, random orientation, and torsion, and the dock conformations are sorted in order of increasing energy. So the lowest energy dock conformation is the first frame. So you see the original conformation in the green, which is the crystallographically determined conformation, overlapped with the dot conformation of the same molecule by autodock. So the lowest energy dot conformation of the benzene, you can see this over corresponds very well with this conformation. So this highlights the power of prediction prediction power of a molecule. And so this is very useful in terms of, say, if you have a, say, for example, you have a database of, say, 1,000 molecules. And it is not possible to see how these molecules interact with the protein by experimental methods like X-ray crystallography. You cannot do a co-crystallization of each and every molecule. So suppose if you have suppose you have a good tool which can help you in predicting how only a top 10 or 20 or 30 of molecules could be a better binder, then you can use them for experimental assay. So this is how this highlights the power of understanding the interaction between the protein and the ligand. Now, if you look at the protein structure-based drug design cycle, you start with a disease of your interest. You make, based on the pathophysiology, genetics, or whatever components, you study the disease and you find out what are the various pathways that are important for the disease progression and viability of pathogen in case of infectious disease and so on. So make a choice of a target protein. The protein structure is studied by either experimental methods, as we discussed earlier, either it is a crystallographic, NMR, or homology modeling. Once you have a structure of a target protein available to you, you can simply do virtual screening in the active site and identify which of them are showing as a good binder, and then these can be progressed further during the, can be taken further up, looking at the protein ligand interaction for testing. Another way is you can simply look at the complementarity of the you can look at the active site and see how a complementary molecules can be designed. And this is what we call as a de novo approach here. By you can simply design a molecule using various linking fragments and drawing into the molecule, which can be further, once you have a synthetically feasible molecules that can be synthesized and tested. So once you get some activity out of these ways, you can further look at the complex of a protein ligand and see if there is a possibility of further improving by replacing, developing further analogs of these, redesigning and improve affinity, specificity, pharmacodynamics, toxicity, accessory. And so this whole process goes on sort of iterative fashion until you get a very good activity, and this goes further down into the drug discovery pipeline. Now, the target was one aspect. The other aspect of this is how, from where do you get these molecules to screen? Now, so at the same time, the chemical databases also have progressed over past 20 years or so, from being mere a repository of compounds that are synthesized within an organization to being a powerful research tool for discovering new lead compounds. So, for example, in CDRI, where I'm working, we have a good team of medicinal chemistry, and we have a whole knowledge base of chemists who have been working here for the past 50 years or so. So all these molecules, the technical knowledge has been deposited in what we call as a CDRI chemical library, and this is also it becomes an asset for the organization as a good research tool. The growth of chemical databases in drug discovery also has research fueled a growth in commercial software for chemical database management and also the publicly available chemical databases. So some example of these databases include Zinc, which is a free database of commercially available compounds, which can be used for virtual screening, contains over 230 million purchasable compounds in ready-to-dock coordinates and 3D formats, and also have 750 million purchasable compounds that can be searched for analogs in under a minute. And this is provided by Irwin Scheuker Laboratories in the Department of Pharmaceutical Chemistry at University of California. Other examples, maybe you can get molecules from PubChem, is 3 linked databases from within NCBI or a drug bank, a unique bioinformatics and cheminformatic resources that combines detailed drugs. their chemical, pharmacological, and pharmaceutical data with comprehensive drug targets. Or you can also have a chem bank, e-molecules, and besides this, you have chemical repositories like commercial repositories like Maybridge or Cambridge, ChemDiff, which provides you a good collection of diversified molecules which can be easily screened, and if you find them having showing good activity, can be easily purchased for biological validation assay. Also, at this point, it's important to mention about now, one of the method was you simply have a protein target and you can blast whole library against the protein target and see from that database can you identify some. But also there's an option to put on some filters before you go ahead for studying the protein ligand interaction. So that way you can do some sort of database searching to filter out only molecules that are of your interest to further take up into the protein ligand interaction analysis. So one first is the exact match search, as the name suggests. This is the most simplest guide. You look for a given fully specified chemical compound in a database and use to find out whether a proposed new structure already exists in a database and so on. Secondly, you can do a substructure search. This is a most common search technique. You copy or paste a set of pieces of chemical structure and the system returns a set of compounds that contain the piece. So suppose you want a molecule that have only a benzene ring, so you define there's a substructure, and then the database will throw back at all the compounds that have that substructure, the benzene in the molecules, and so you can now screen that subsets of molecules having benzene for further interaction analysis. Search involves mapping of all atoms in a query structured against those of candidate structure from a database. While the third and the most important, in my opinion, is what we call as a pharmacophore search. Now, pharmacophoric or a three-dimensional substructure searching involves a set of atom bonds or groups that is combined with a specific three-dimensional constraint. And these three-dimensional constraints could be distances between these pharmacophoric groups or a defined angles between the pharmacophoric groups also. So the pharmacophoric groups could be hydrogen bond acceptor or a donor or aromatic ring or a base or hydrophobic groups and so on. Now, since there are so much constraints that are being put into developing this query for pharmacophore search, the process is generally much slower than the two-dimensional substructure search, because here you require to examine all the XYZ coordinates for atoms of the candidate structure to compute all the restraints that have been put on the query. But this is important because this can provide you an indication of whether a set of structure can bind to a receptor or a. So the chances of getting a good binder using this search is more as compared to a two-dimensional substructure search and so on. So for example, suppose you have a high quality three-dimensional structure of a target bound to a known ligand. So you can see how bound ligand interacts with the protein. So you can simply extract those features, those pharmacophoric features, and then use this to develop a sort of pharmacophore query, which you propose as having these groups, and also you define that I want these distance constraints between these groups. And once you have this constraint pharmacophoric ready with you, you can use them for screening a library. So these are some example of molecules from SNX library or Maybridge library that have been found to be extracted, which have the similar features as having defined in the pharmacophoric model. This is being developed using a high quality 3D structure of a target bound to a known ligand. Now, how this has been, Dr. Yatendra, I'm clear and audible?
Yes, sir.
Yeah, okay. Uh, So actually, there's a problem with the online teaching or interaction is that you don't know whether on the other side people are listening to you. So you have to keep on checking in between whether you are still connected or not.
We
Okay.
understand that, sir.
So, yeah, yeah, yeah. Okay, okay. So,
Thank you very much.
okay, okay, yeah, okay. So, so we started using these methods of virtual screening way back in 2004 or 5 when started developing models of virtual screening to identify hits. Some of the very earlier example of this was where we used ligand-based virtual screening uh, uh, for identification of chorismate mutants. This is a very important enzyme of Shikimate pathway in mycobacterium tuberculosis, where we screened, we built a pharmacophore using substrates of chorismate mutates, and this was used to screen a subset of CDRI molecules of say around 15,000 molecules out of the total maybe 50,000 odd molecules. Or these led to the identification of 95 hits based on the proposed query. Of these, further looking at the interaction analysis between the protein and ligand, we picked up 15 molecules for actually taking into the biological assay. Now, this approach is important here because these molecules in a given library of any organization, these are being synthesized for various other disease areas. And so here this brings in the concept of sort of repurposing, which is very much useful in current application of drug design, where you identify the known scaffolds which have been developed for some earlier diseases and have been uh, dropped out because of some reason or other. That can be repurposed here for putting into use for some other disease area. So in this case, we put up a screening and identify 4 active compounds which show a good activity in biological assay against the and so once you find some active compounds, you look at the interaction between the protein and ligand and see how these can be further optimized. And this went into the optimization cycle. Uh, the advantage at being CDRA is that we also have a good team of chemists, so you can discuss with them, tell them, okay, this is the molecule, this has to be optimized in such a way that the potency of that particular molecule can be further improved. Similarly, so this was done by Ashutosh. He was one of my earlier PhD students. So later on, he worked on another protein called uh, thymidylate kinase. And this is an important enzyme which catalyzes the reversible phosphorylation of DTMP to deoxythymidine 5' diphosphate in the presence of adenine ATP. So he screened a Maybridge database and also developed a special, a unique method of prioritizing. Once you get screen out a molecule, you can. He developed a method called structure interaction fingerprint, where which is nothing but a one-dimensional representation of a three-dimensional interaction between a protein and ligand. So this can help you in further prioritization of a molecules, which led to the development, identification of good low microgram per ml hits, and these were further set into optimization cycle for in a sort of iterative cycle in collaboration with medicinal chemistry. And so this was, I'm just talking about the initial lead that we got. I'm not discussing about what went further about what happened to this molecule in the later stages after the optimization. Similarly, Nidhi also targeted another protein called from Mycobacterium tuberculosis. This is a PKNG. This is a serine threonine protein kinase that plays a crucial role in regulatory process within the mycobacterial cell and signaling cascade on infective host cell. So, based on our own molecules from our chemistry colleagues, we developed a uh, pharmacophore models, and this was used to screen a Maybridge database, which is a collection of around 55,000 molecules. Um, Here, she used a set of three different docking methods and tried to find out which of the molecules have a consistent screening, and then this gives the better understanding of interaction of the potent molecules with the, and this way six molecules she identified in a biological evaluation against this protein assay, and three were found to access significant inhibitory activity against, and that show really to kill the bacteria also. In another disease area, so that was in Mycobacterium tuberculosis related to tuberculosis. Shagun worked on pharmaco-based virtual screening to identify human DNA ligase 1 inhibitors, and these have therapeutic relevance in the area of um, and developing anti-cancer agents. This joins Okazaki fragments during DNA replication, and its inhibitors are likely to be anti-proliferative. So this includes a set of known molecules of DNA ligase that was taken from the literature, and that was used to developing a pharmacophore model based on the common features that were there in the known inhibitors of the protein. So here she used a GASP and unity of Sybil that was at that time, and that's after screening out top 3,000 compounds were docked to the human DNA ligase. Five were selected for DNA ligation assay, and finally two active compounds were identified to show a very good like and DNA ligase inhibition activity in a biological assay. In another example, Vikas also used this method of virtual screening to identify real molecules where he targeted CHEK1 in serine threonine kinase, which is involved in the regulation of cell cycle checkpoint. So the crystal structure of CHEK1 was used which was in a bound form, a co-crystallized structure of CHEK1 with protein thiopyridine derivative. So the special features of the molecule were simply extracted, and this was used to develop a pharmacophore model, which is a 4.3D pharmacophore model having two hydrogen bond acceptors, donors, and aromatic ring, and developed by Unity and Sybil. So this was screened using, so the database that she used was a Baybridge. And finally, after all the filtering and analysis, she took 5 hits for actually taking into the CHEK1 kinase assay and also at the same time yeast cell growth assay. And 4 compounds were found to show a good IC50 value in the range of 4 to 13 micromolar. So the idea of this is not to directly hit the jackpot and say that after screening that I have found a real molecule that can simply directly go into the clinical trial. The idea is to identify some in a more rational way to identify certain molecules that can develop as a lead molecules and then can further optimize. So this helps you in having a more focused approach in developing a particular chemical scaffold that can be taken into the later stages of. So some of the recent trends that we have been observing and have been applying recently in our projects also is how do we integrate artificial intelligence or machine learning, deep learning basis methods with virtual screening approaches in drug design. We have seen that with the rapid explosion of genomics and chemical big data from STS and combinatorial synthesis, AI approaches, including both the machine learning and deep learning, have become an indispensable tool for drug designers to mine chemical information from large compound database to design drugs with important and precise biological properties. Now, as compared to the physical models that relies on explicit physical equations like quantum chemistry or molecular dynamic simulation or machine learning approaches. These uses pattern recognition algorithm instead of explicit physical equations to find out mathematical relationship between empirical observations of small molecules and extrapolate them to predict chemical, biological, and physical properties of novel compounds. And this integrated approach that have been very useful have shown a very good results. Uh, some of the works that have been taken by one of my other PhD student Shagun in past 4 or 5 years, where she tried to integrate all these machine learning methods with the virtual screening and and try to put in use into various targets for anti-cancer development of anti-cancer agents. So some of the targets that she worked on included the DNA methyl transferase or DNMT1 as we call. It has been shown that this particular protein inhibitors can suppress tumor growth and have significant therapeutic value. So here what she prepared, she used a huge collection of data set, inhibitor data set of DNMT1 inhibitors, classified them on the basis of training and test set using uh, 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 developed a farm for models, and this is the models were generated, validated by various methods, virtual screen. Once she get a set of molecules which have found to have which were found to map to the pharmacophore model, she applied some sort of machine learning classification in the form of Bayesian classification models or ensemble based docking, and then these further helped in prioritizing and shortlisting the compounds that had a good probability and chances of giving good starting leads. So starting from say around 50,000, 3,000 molecules, she was able to actually get a good inhibitors which have shown a good activity in biological assay against this particular protein target, and three DNMT inhibitors were identified. Similarly, in another HDAC inhibitors, which she worked where she tried to integrate these machine learning models with the virtual screening approaches, is the targeting of hashtags, which this reverses the acetylation by removing acetyl groups from the lysine residues. So here you use a S support vector based machine learning based classification method to classify the data sets, and then use the best SVM models to screen out pharmacophore based models. Uh, uh, and these are further used by docking-based filters to identification of three novel HDAC inhibitors, which have shown a very good activity in the biological assays. In another targeted method where she used a histone acetyl transferase as a target for developing models for identifying inhibitors of histone acetylide transferase. These have very important targets for treatment of for developing of anti-cancer agents. So with the information of known inhibitors, she designed a study of identification of new inhibitors using multiple machine learning techniques. So uh, the data set was further classified into training and test set. Descriptors were calculated, models were generated using KNN or naive Bayesian random forest or SVM. The best classification model was used for screening. Naive databases, consensus were identified, these were docked, and finally she zeroed in down to the selection of most suitable hits for biological evaluation, which resulted into the identification of active hits in the screening of these molecules. So these are some of the examples from my group, but there are many examples that have been cited where actually instead of simply identification of lead molecules, they have went on to develop real molecules that have shown a therapeutic utility. For example, some of the very important example of structure-based drug design includes the development of captopril, or which is used which has been developed by targeting angiotensin converting enzyme used for treating hypertension and developed by SQUIP, or by HIV protease inhibitors like lopinavir or nalfinavir. These are essentially antiviral uh, compounds have been developed by Abbott laboratory, or nalfinavir again by Eguron or Lilly, or one of the very important enzyme like Relenza, which is essentially a zanamivir. It is antiviral again used to target influenza and neuraminidase, developed by a collaboration with Monash University and Glaxo Wellcome. And so here lots of structure based efforts have been put into the development of these success stories. Um, so I'll end with some of the interesting facts, like over 90% of the drugs that enter into clinical trials fail to make it to the market. This is harsh, but very the harsh fact, but it's true. Average cost to bring a new drug to market is estimated to be more than $800 million, and this is a huge amount. But the analysis of pharmaceutical analysis of past 15, 20 years, it has shown the impact of computational modeling or drug discovery. Well, it has been observed that the use of computational modeling in modern drug discovery, it has actually speeds up the key steps in drug discovery process by combining various aspects of structural and bioinformatics, structural biology, structure-based drug design, and so on. So the application of bioinformatics, cheminformatics, molecular modeling in drug discovery and development is expected to reduce the annual cost of developing a new drug by 33%, and the time taken for drug discovery by 30. Essentially, the drug discovery cycle, if you recall, it's say around 10 to 12 years. But if you put these efforts also, it reduces the time by 30%. For example, the HIV protease inhibitors, they come into market by mostly in 6 to 7 years. So once you reduce the time, you reduce the money also that goes into this. And this is what is the importance of the role of computer-based drug design, which also includes the structural bioinformatics, structural biology, structure-based drug design into the this very important process. Well, we can say today the discipline of computational medicine has begun to mature and is routinely used component of modern drug discovery process. So I end at this. I would like to thank you all for your patience to hearing me out, and hope I find this is useful and goes well with the theme of faculty development program in this very important area of this. So if you have any questions, I will be happy to answer. Thank you so much.
Uh, Thank you very much, sir, for such an informative talk. I guess we all felt so lucky to have got this chance to listen to your enormous experience. Sir, you have made us all believe that research is to see what everybody else has seen and to think what nobody else has thought. I would also bring to this in record that you have explained all the topic in such a convenient and interesting way, keeping in mind the various background of the participant, that everyone has felt the depth of the talk. The topics covered by you, like utility of modeling, retrieval of 3D structure, structure-based drug design, molecular docking, and others. Sir, from the core of our heart and on the behalf of NSUT fraternity and all respected participants, I would once again like to thank you for sparing your valuable time and enlightening us all with the insight of all the knowledge. I would now ask Dr. Sonika Bhatnagar, HOD ma'am. Ma'am, please say a few words.
Thank you, Dr.
Actually,
Katani.
more than saying words, I have lots of questions. I know there are very
Yeah,
nice
sure.
questions from the audience also, but to Dr. Imran, as somebody who has worked with a wide variety of drug targets of all kinds, different diseases, Do you find that infectious diseases are significantly different from lifestyle diseases? आपका क्या experience रहा है इस बारे में?
Actually, I think that principally, actually, they have to be similar. Approaches are similar, but there are very basically fundamental differences when we compare the development of lifestyle disease like a cardiovascular disease or maybe a hormonal disorders, other like diabetes or so on, as compared to the infectious disease like say a malarial parasite. The malarial parasite, actually there's a lot of problem with the assays that are not there in the parasitic diseases like malaria or tuberculosis, specifically the target based assay. So here in this case we have to rely on the in vivo assay or the cell based assay more as compared to the specific in vitro target based assay. In case of course, we have a very good models in lifestyle disease also, but also the specific drug in vitro, say a protein-based assay also. This is one of the basic, most important limitation or the problem that we face when we design drugs for lifestyle diseases or infectious disease. But otherwise, principally the methods, you have to apply the same, look at the exploit, and essentially you are exploiting the structural information of the targeted proteins or for other macromolecules. And so if the methods are same, it should yield a good results.
Okay, Atender, please go ahead. Participants have lots
Yeah,
of questions.
so like there are a couple of questions from the participants, so I will take them to you by one.
Yeah, doctor.
There is a question from Sir Dr. Aminul Manan. How to know which is the best key with respect to drug target, sir? Because several keys can open the lock, and what are the parameters?
Okay, so suppose now you have say a bunch of keys. Okay, each key is a different in shape, in size. So you have to find the best key which goes and fit into the clock. So each, if you look at the each key, it has a different maybe the designing is different. लेकिन आपके जो लॉक में जो लीवर्स होंगे वो स्पेसिफिकली डिजाइंड उसी के लिए होंगे. Similarly, in a molecules also, you have a different functional groups. And if you have to get a good efficacy or a good binding with the targeted protein, they should make a very good complementary interaction with the protein. So not all molecule having different features will have a similar binding. So the best binding, which is more optimized, could be your best key for your lock. And this is evaluated by, of course, various methods are there to see how interaction in terms of scoring function or whatever interaction that you see can help you in prioritizing which is going to be a good key. Some of the keys may simply you have to discard it, but some may fit but cannot rotate. So you have to little bit tweak it to see if it can be further optimized to get fit into the log. So this is how we do a building of analogs. You have a good molecule, but it does not show activity. So you do try to build some analogs that can have a better activity against that target.
There is another question from Mohammed Amir. What should be the criteria for the selection of best dog structure when we get various conformations with little difference in binding energy?
So actually binding energy, you cannot always rely on the binding energy because sometimes you get a very similar exactly binding energy. So the best way is to look at the visual inspection, try to see how the molecule is interacting with the, and suppose if you have a reference structure also, so for example in a co-crystallized structure, you see what are the important binders that play a role in binding of this, what are the specific pharmacophore groups that are responsible for affinity, giving the affinity of molecule. So you focus on that. You see, you have three molecules with a similar binding. You can check visually whether they are making good interactions that are desired to be having binding that are desirous to be there for showing the interaction with. So you can select them on the basis of those particular. So manual intervention is also a very valuable tool.
There is another question from him, like, do all the molecular modeling softwares give similar output results, or like which software one should prefer? So any
No, essentially, as I mentioned, all the molecular docking models, they generally calculate how a molecule gets binds to them. They have a different algorithms. They use different methods. So that's why we also try to take into account. So some methods may use different calculation algorithms. Some may miss out on maybe not focusing on hydrophobic or hydrophilic or what some sort of. But the best way is if you have an access to 2-3 programs, then you try to do that to see if you are evolving some sort of consensus scoring, and based on that you can see, you'll try to see if you can select or shortlist them of also as a taking forward. So, but I believe that most of the programs take care of how good or bad binder could be there when you look at the interaction between them.
there is a question from Lakshmi Shanmugam. Is there a way like you can activate or inactivate proteins, sir, like using drugs?
Activate inactivate protein. So in that case, so you have to look at the functionality of the protein. If you say that you are not looking, you are not finding inhibitor, you want to look for say an activator or an agonist or not an antagonist sort of thing. So you in the binding of a molecule, if it is an activator, if it binds to the site, it is going to activate the molecule, the whole protein. So it depends on whether you are planning your experiment as an inhibitor or an activator. Yeah, if you are designing an activator for a program or for a set of molecule, this should function as an activator of that particular molecule. So many proteins require an activator, activating a activity of a protein to be a good, to be used as a good target and as a drug. Yeah.
There is another question from Priyadarshini. How reliable is the result of Autodoc, sir? As it is a freeware, can we rely on the results of Autodoc and go for the wet lab work?
Sure, definitely. It's a good program, and that if you recall that in my example of Autodoc, which I showed here, instead of showing other programs, I showed only the Autodoc video. She can also visit that. Unfortunately, that animation was not working in this, but she can go to Scripps Lab and find out the screening. These are actually very well validated methods, and can be relied very well on the prediction that can be. Of course, it is not always that you think that हमने 10 मॉलिक्यूल आइडेंटिफाई किए एंड 10 के 10 होने चाहिए, ऐसा कभी नहीं होता है. Our experience that suppose after carefully filtering out the molecules, ultimately if we took 10 molecules to the experimental, we mostly end up with 4 or 5 good activity. So the idea is that you rationalize in a such a way that you are finding, अगर आप 50,000 मॉलिक्यूल से शुरू करके अगर 3-4 मॉलिक्यूल के ऊपर भी if you are finding it showing good activity, then I believe that is a good success for your experiment.
There is another question, I think, which you have partly answered, like with respect to like why do we get different scores in different molecular modeling tools? I think this is again depending on the parameters.
Yeah, the parameters and how the program calculates the interaction.
The next question. There is a like in publication, like for publication point of view, which molecular docking tool can be preferred? Like at times, like there is question like you specifically use this one only, then things get published. Otherwise, freewares at times they are not accepted, sir.
No, I don't think this is a good assumption. Autodoc is a freeware, but you find the plethora of lots of citations of Autodoc there. So this is, if you have a good access to Autodoc, this is, I think this is one of the good programs. This is, I again tell that I told about this because many of the faculty colleagues that are participating in this may not have access to the commercial programs. And that's why I try to tell them this is a very good program, can be used very good. It can give you, it depends on if you use it properly with the proper protocol, you can rely on the results and can be published without any problem.
Like how do you differentiate between cis and trans isomer of compound while docking? Like what are the main points one should consider while doing this study?
Yeah, it depends on जब आप stereochemistry आप define करते हैं किसी भी molecule की उसमें, so while drawing a molecule you define whether a molecule is in a cis or trans. So this is very important because sometimes a molecule could be a mixture of both cis and trans. So isometric, it could be a racemic mixture also. So there are programs which can tell you what is going to be the stereochemistry of a particular, which particular point it has to be taken care of while designing whether it's a cis or trans conformation for a particular molecule. And most of the the modeling programs can take care and tell you how to do this. So that's not a should be taken care. I think she can look into that aspect.
So like there is a question from Pawan. Is it necessary to do energy minimization of protein like every time when you are doing docking, sir? So when like through
No, if you are starting with a very good high resolution protein structure, so there is no need because the resolution itself is very good, so you can directly take it. Now, suppose if you are using a homology model or a model structure, but mostly of programs generally take care of minimization when they give you an output. But suppose if you do a Ramachandran plot check and find okay there are still constraints, then maybe you can do some sort of energy minimization, try to minimize it and to release all the steric constraints that are there in the protein, and this can be used further for taking up further for maybe protein ligand interaction studies and so on.
there is another question. I think you have partly answered in this one itself. Like how to optimize protein structure and understand that it is a good. It is the structure we obtained is a good one, and like the one basically which has been modeled is very close to like the native
Yeah,
conformation.
of course. Once you model it, you can have a Ramchandran plot. I mentioned Ramchandran plot is a very good program to assess the quality of what if or ProCheck. These can be used. This can give you indication whether all the residues that are part of the model are in the allowable region or not, and this give you indication that the molecule is the model protein is good to be taken care at the next step of your
Is
experiment.
there other than like something with respect to Ramchandran plot, like higher end versions? Because the candidate asking like rather than Ramchandran plot, anything else which can be used to be more
Why
specific.
they are not satisfied with Ramachandran plot?
Ramchandran.
I think Ramachandran plot is a gold standard in this. If they are not satisfied with this, I think so. If it is not showing good result, I think there's some problem with the protein structure. Can be further relaxed or minimized and try to see if there is some problem with, or try to optimize it. I think Ramachandran plot will give them a good indication.
Like it is a very interesting question. Like if a standard drug is binding to a receptor in a to particular amino acid, is it mandatory that the design analog has to bind to the same amino acids to be of equal efficacy?
Okay,
If
it
like
is an
standard,
active,
yeah,
if it is an active compound and it is shown to bind to that particular residue,
yeah,
then while designing, you try to conserve that interaction in addition to try to increase another interactions. So if you break that interaction, maybe you are going to lose that activity. If it is important, or if if the co-crystalline structure also tells you that that interaction is important, then it is advisable to retain that conserved interaction in your design of analog of that molecule.
so like this is the last question, sir. So
No.
Lakshmi is asking like I think it's more of a technical question. She's getting lot of error in the autodoc when she's trying to use sample compound provided by them in the tutorial room. But docking occurs
Mhm.
when she uses DSC or PYRX. So she has looked
Mhm.
for various websites and also written to the Autodoc regarding this. So what is the issue? Like it's not working for her. Maybe sir, like I think if you read it in chat, it will be much clear to you. If chat box is available to you, I think the question will, this technicality will be much more clear then.
Oh, I understood that she is getting some sort of error while running this script. I think that's
Yes, sir.
well. It is very difficult to comment on what exactly is the problem unless we see the whole the output of that. But maybe it is. I think it's an operational error. Maybe she might have not some of the libraries are not being activated, or some parameters are not being set. Uh, um, uh, I think that could be the probable reason of this technical error, but she can always get in touch if they have any issues individually. Maybe we'll try to sort out any for any faculty member who has any question query regarding any of the aspects of computer can freely contact us. There is no issue with. We can try to discuss and sort out any problems with that.
I think the person like who asked regarding Ramachandran plot, he said like he doesn't want it. It is the reviewers who want something else other than Ramachandran plot. So what to do? How to satisfy the
Okay.
reviewers?
Okay, so that
You can try
that
pro check also.
yeah, pro check what if. So essentially, these programs they essentially do the Ramchandran plotting, but maybe if he or she can write that you did it with pro check, or what if instead of writing Ramchandran plot, maybe it can work.
I would try to understand the reviewers' concerns. They might be, because reviewers generally don't ask for specific
Mhm.
tools. They generally have some other underlying concern that you have not addressed. So maybe you should look at that.
Yeah, probably.
So like now there are no more questions, and with this I would like to thank you so much for such an interesting and interactive talk, sir. And
Thank you, Dr. Yatindra. Thank you, Dr. Sonika. It was nice interacting with you, and it is indeed a real pleasure for me to be part of this FDP program. And thank you so much again.
Thank you for your
thanks once again, sir,
participation.
like for taking time from your busy schedule. Sir. Thank you so much.
Thank you very much.
So, participants, we will be starting our next session at 1:30.